To start this video, it's essential to know what is a chemical sensor. We can define a chemical sensor as those devices that detect and convert chemical information, such as concentrations, pressure, and activity of particles, into an electrical signal to obtain qualitative or quantitative time-spatial revolved information about specific chemical components. Once we know what is a chemical sensor, it is important to mention another fundamental definition to understand the specific chemical sensor that we are going to explain later. So, this definition is about chemiluminescence, and it is basically the emission of light from a chemical reaction. But the real reason to mention the definition of chemiluminescence is in order to understand the phenomena called as chemiluminescence resonance energy transfer, or CRET, and this is a phenomenon generated from a chemical reaction rather than a photon absorption with external excitation. Besides, CRET is a free autofluorescence interference, and the setup can be simplified to achieve an extremely high signal to background ratio for sensitive detection. Now that we know these important terms, it's time to start talking about this chemical sensor and its capability to detect miRNA. This detection of miRNA is possible because the chemical sensor used in this investigation was based on a new type of CRET based on mesoporous silicon nanomaterials. So, how does this chemical sensor works? Well, the CRET energy transfer distance between the donor, that in this case was horseradish peroxidase luminol H202Cl system, and the acceptor was a dye-labeled DNA which was controlled by a DNA duplex. Therefore, the target miRNA was sensitively and selectively detected based on the change in donor emission after hybridization to the target domain on linker DNA. With high loading capacity, high energy transfer efficiency, sufficient delivery, good biodegradability, and low emission interference, CRET-based imaging of miRNA was employed in mice. The first step to prepare the chemical sensor is to synthesize the mesoporous silicon nanomaterial following the next instructions. First, 8 milliliters of water and 8 milliliters of hexadecyl trimethylammonium bromide were mixed under stirring at 32 degrees Celsius. After adjusting the pH to 8 with a sodium hydroxide solution, 700 microliters of tetraethyl orthosilicate was added into the solution and stirred overnight. The resulting mesoporous silicon nanomaterials were collected by centrifugation washed with ethanol. Once the mesoporous silicon nanomaterial were synthesized, it was functionalized with NH2 on its surface using 10 microliters of 3-aminopropyl triethoxysilane dispersed in 4 milliliters of ethanol. This mixture was added into the mesoporous silicon nanomaterial solution, and the reaction was carried out for 3 hours under stirring at room temperature. The obtained NH2 modified mesoporous silicon nanomaterials were dispersed in water for further use. The second step is the synthesis of a DNA duplex with the following instructions. Every 50 microliters of DNA duplex solution contained 8 microliters of a DNA carboxyl acid solution, 10 microliters of DEA DNA solution, 5 microliters of PBS buffer, and 27 microliters of DEPC treated water. The solution was maintained at 94 degrees Celsius for 5 minutes before naturally cooling down to 35 degrees Celsius. The DNA duplex was applied to the subsequent synthesis after stabilization for 30 minutes. Finally, the third step is the synthesis of CRET-based mesoporous silicon nanomaterial with the following instructions. First, 1 milliliter of the NH2 modified mesoporous silicon nanomaterials and 50 microliters of EDC were mixed and stirred in an ice water bath. Subsequently, 1 milliliter of HRP and 50 microliters of the DNA duplex were added into the mixture and stirred for 2 to 3 hours. Then, CRET-based mesoporous silica nanomaterials were formed based on the formation of an acid bond between carbonyl groups and amino groups. Finally, the products were purified, dispersed in DEPC treated water, and stored at 4 Celsius degrees. In order to confirm the successful synthesis of the CRET-based mesoporous silica nanomaterials, morphology characterizations were employed, as high-resolution transmission electron microscopy and dynamic light scattering analysis showed an approximate diameter of 20.3 nanometers for the pure mesoporous silica nanomaterials and 26.3 nanometers for the CRET-based mesoporous silica nanomaterials, demonstrating the 3 nanometers thickness of the modified layer. Both the successful modification of HRP and DNase on the MSN surface was further demonstrated by X-ray photoelectron spectroscopy. As shown in Figure 1c, the intensity of peaks at 284.5 electron volts and 401.1 electron volts increased, and new peaks at 129.9 electron volts and 711.7 electron volts were observed after functionalization. CRET-based MSNs also exhibited remarkable UV-vis absorption at 260 and 400 nanometers, that is attributed to the absorption of nucleic acids and HRP, respectively. Additional confirmation was made by BAT analysis, showing lower isotherms for functionalized MSNs than for bare MSNs. The step mass loss recorded by thermogravimetric analysis also demonstrated the successful loading of the HRP DNA duplex on the MSNs. Therefore, CRET-based MSNs were successfully prepared for subsequent miRNA detection. Furthermore, zeta potential changes during the process of preparation confirmed the successful synthesis of CRET-based MSNs. The in vitro detection of MIR155 by CRET-based MSNs was employed in real human serum. After the 15-minute incubation for strong displacement reactions at room temperature, CL signals at 430 nanometers were recorded. Figure 3a shows the CL emission increased with the increasing target concentration from 4 to 20 pm. The inset of Figure 3a demonstrates good linearity for CL emission as a function of the MIR155 concentration, resulted in a limit of detection of 2.2 pm, much lower than other CRET methods. Additionally, CL imaging without any excitation showed increased CL emissions with the increasing target concentration. Therefore, CRET-based MSNs can be used to detect miRNA in complicated biological samples. Specificity was examined by comparing 3-1 base mismatched DNAs. As shown in Figure S10, only weak CL signals were recorded during the detection by CRET-based MSNs, which was due to the failure of strand displacement. The selectivity was further validated by the detection of other liver-related miRNAs. Therefore, CRET-based MSN detection showed high sensitivity and specificity for the detection of MRI-155. Finally, the in vivo imaging of MIR-155 in HepG2 subcutaneous xenograft nude mice was performed at different tumor stages. Figure 4b shows increased signals of MIR-155 with increasing tumor stages. This result primarily confirms the possibility of in vivo detection by CRET-based. This work has served to expand the development of CRET-based techniques for the sensitive detection of biological molecules. CRET-based MSNs proved successfully to be highly sensitive and specific for the detection of carefully chosen miRNAs. As demonstrated, CRET-based MSNs exhibit great results in miRNA detection in human serum and mice, although further studies are still needed to improve the sensitivity of the present method for in vivo imaging.